Most bioinformatic analyses are going to be considered as preclinical. That means it is based on data generated in lab experiments or retrospective studies. To explore these, let's look at examples of cell lines derived from patient tumors in oncology, animal models of human disease, and explore how these can be used alongside databases on various specific diseases, for example, TCGA in oncology. Then we will discuss how such insights can be validated and what considerations they bring to biobanking and clinical trials. Next, technologies and experiments are already affecting research, hospitals, and are used by pharmaceutical industry to design specific treatments and adapt them to the needs of individual patients. A lot of times, not all of these experiments are possible on real patients. Instead, we can select tumors in different types of patients, produce those tumors as immortalized cell lines, and use those cell lines to understand the basic biology. Unfortunately, that biology cannot be fully studied just using cell lines, because cell lines don't have all of the elements of the system or the whole body that actually functions as a whole. And so in many cases, in oncology, instead of just using cell lines, there are also tumors that could be extracted in full from a patient, grafted into an animal model, and then studied in terms of their interaction with the rest of the body. Ultimately, such experiments can provide better insights into biological factors that are relevant for patients, and different types of tests could be first performed on these models before we actually develop a clinical trial where such treatments or diagnostic procedures are used on actual patients. This is also changing the way that we apply big data analysis techniques or bioinformatics in real-world clinical needs. So typical process is to collect, integrate, and analyze big data coming from a variety of sources and ultimately change the way that this can be used in a big data approach for precision medicine. We can see this oftentimes used when large-scale data is being collected on a particular disease. A good example is the Cancer Genome Atlas. The Cancer Genome Atlas is a work performed over several decades to collect samples with multi-omic data on patients and use their clinical phenotypes to find important associations. The cancer genome data is available through the Genome Data Commons portal that allows us to find multi-omics datasets with clinical information about each sample, which gives a much better understanding of how cancer variation is diagnosed and treated and how successful those treatments can be. These large-scale datasets are continuing to be collected, and today we have the All of Us dataset as well. The All of Us research program, which is led by the National Institutes of Health, is building one of the largest biomedical data resources of its kind. The All of Us Research Hub stores health data from a diverse group of participants from across the United States. Researchers can access All of Us data and tools to conduct studies to help improve our understanding of human health. Now, it also provides us with a much better understanding of what we call healthy, or the variation of healthy individuals that can be used as a reference when we study subgroups of patients or patients that are of different ages, sexes, ethnic backgrounds, and socioeconomic status. In addition, there are projects that are aimed at characterizing the healthy condition and preclinical variation. One such example is the NIH Human Microbiome Project that allows us to look at different types of microbiome perturbation that is associated with different types of diseases. These include depression, autism spectrum disorder, cardiovascular disease, asthma, arthritis, cancer, and diabetes. By understanding the microbiome diversity, composition, and changes associated with different types of phenotypes, we can start seeing the impact of the microbiome on diagnostics, but also treatment, because patients with different microbiome can metabolize treatment at different rates. Now that we have reviewed the rationale for bioinformatics and NGS in biomedical research in the clinic, let's review case studies of clinical research with real data. These include oncology, neurodegenerative diseases like Alzheimer's disease, and infectious diseases like COVID-19, malaria, or Ebola. We can look at how viral genomics and evolution can help us understand the spread of a disease and identify variants of concern in pathogens or genetic predisposition of some populations to be more susceptible to acquire disease and potentially have worse outcomes. We can look at immune responses, where we characterize gene expression and study biomarkers or genes that are associated with a particular response. And sometimes we can look at microbiome and see how microbiome plays a role in disease progression, drug metabolism, and also predisposes certain people to different clinical risks. On the OmicsLogic portal, you will find a number of case studies and curated datasets around these different topics. Some of them are based on full analysis of datasets that are already published. Simply by following the steps described in these case studies, you will be able to make important discoveries that today are being used in the clinic. Some of them have to do with drug metabolism, identification of somatic mutations in tumor development, inherited or germline mutations like the sickle cell anemia, rare genetic diseases, as well as drug resistance associated with different types of antibiotics and pathogens. Oftentimes, just by reading the abstract, you get a good grasp of how bioinformaticians can use such data to model precision medicine. For example, on this project, modeling precision medicine of breast cancer, you will find how researchers use machine learning to study transcriptomic data and identify subgroups of individuals that are going to respond differently to different treatments. In the study, researchers use multi-omics data integration to look at different types of molecular profiles, integrate them, and compare them to reference databases like the TCGA to understand response to treatment to drugs that are either already used in the clinic or being studied in clinical trials. To continue learning, we invite you to explore these associated resources for the lecture. We will go over the coursework, the case studies, and other examples from literature. To benefit from today's lecture, you can select one of these to refresh your memory of basic terminology, learn more about the various techniques, or practice what we have learned with curated datasets on the topics that we covered in today's session. The contents of the session correspond with practical lessons that you can complete independently that are available on the OmicsLogic Learn portal. With the full course, specific lessons that are of relevance to today's session include Lesson 2, Bioinformatics in Healthcare from Course 1, Introduction to Bioinformatics. In this lesson, you will get an overview of examples from healthcare area where bioinformatics can be applied. You will also see examples and videos of medical professionals discussing bioinformatics in precision medicine applications. Another good course to look at is Bytes and Molecules. This course is a great place for those who was confused by a lot of the terms that we used in today's lecture. Anyone that would like to review the basic terminology and get a good overview of topics that we cover in this course should look at this course in full. But specifically, we looked at this lesson 6, called Introduction to Genomics. If you are interested in genomics, you can also look at these additional lessons in the Course 3 course called Genomics. In Lesson 2, you will see how you can work with DNA sequences in R, and in Lesson 3, you will get an introduction to large-scale genomics. The full course contains a lot more information that you can choose to explore independently. Some of these will be covered in the future lectures, but do not worry about completing them for now. At the end, you will also find examples that I already mentioned. These examples include such projects like Ebola, immune response changes in cancer, modeling cancer precision medicine, or tuberculosis infection and treatment, finding drug resistance variants. So let's recap what we have learned so far. First, we learned how combining clinical and molecular data helps reveal underlying factors contributing to the disease, which further helps us provide a precise diagnosis and effective treatment to the patient. Further, we learned about next-generation sequencing technologies for genomic, transcriptomic, and metagenomic data that provide important insights for precision medicine applications. We also discussed the role of bioinformatics in taking research results from the lab to the clinic to benefit patient treatment and diagnosis. Finally, we introduced a set of case studies in precision medicine, from inherited sickle cell anemia to drug resistance in Mycobacterium tuberculosis. In the next session, session 2 on genomic data analysis, we will go one step further to better understand what is genomic data, how do you perform variant calling analysis, what is the difference between germline and somatic mutations, and how to determine a variant's clinical relevance. At the end of session 2, you will have a thorough understanding of genomic data, variant calling analysis, as well as specific techniques for clinical annotation of variants in cancer.